Swayam Prabha. Digital India. Educated India.
Welcome back to the fifth lecture of the third week, which is the last lecture of this week. So, let us have a little recap what we did in the last 4 lectures. So, this week we have completely devoted on the muscle, starting from the development of the muscle, classification of the muscle based on force, based on morphology, based on the myosin and actin, which eventually lead to the force generation. And we talked about the hierarchy of the muscle in terms of molecular to tissue organization, and then in the last class we talked about the molecular architecture of the muscle. So, this is the picture where we ended, where we have the sarcomere. You could see the sarcomere out here. You have the Z lines, the red. You have the titin molecules attaching to the myosin filament, thick myosin filament, and thin actin filament, which are shown in red. So, let us start this class with a small recap of the organization of muscle, and after that we will come back to the molecular details of these individual structure. And I told you there is something very interesting out here. The interaction between actin and myosin filaments and generation of something which is termed as power stroke. Okay. So, we are into week 3 and this is the final lecture, the lecture 5 of week 3. So, in terms of the organizational level, this is where we are. If this is what we call as muscle, this is how we should be able to remember. So, we pick up the fibers, the smaller unit of it, the individual fiber attaching together to form muscle, right. From there we pick up another smaller unit, which is the myofibrils. Myo meaning muscle and fibrils, myofibrils making fiber, fine. From the myofibrils we have now narrowed down our search to filaments, thick filament and the thin filaments. So, let us color them differently. Okay. This is the red colored filament and you have these blue colored filaments. So, these red color filament which are thicker in diameter, they form the myosin filaments, and thin color or lesser diameter filaments which are blue in color form the actin filaments. So, if you look at this picture very carefully, you will realize the level of hierarchy what is being followed in nature, from self assembly of some big molecules like actin and filaments, which are proteins essentially, they align in a very unique self assembly to form what we call as filamentous assembly, what we call as myofibrils. These myofibrils then form even much more complex assemblies called fibers, and these fibers eventually form even a much more higher self assembly called muscle. So, what is the central theme in this whole process? If you look at it, there is one word which explains this process, and that is self assembly. So, if you look at it, at every level in nature, there are certain unique rules of self assembly which is dictated to lead to the formation of such complex structure. Okay? And the future lies in understanding these kind of self assemblies, because these self assemblies or understanding these kind of self assemblies will one day make us to dream, or maybe will help us to achieve the dream of developing something called artificial organs, where we will be able to develop structures according to our needs and requirements in the laboratory. But in order to do so, or you can call it in vitro, outside the system self assembly. As a matter of fact, our very core of understanding our own self depend how into the minutest or the finest detail we understand the forces governing self assembly of molecules to cells to tissue to organ. Okay. So, keep this picture as a part of your brain map. Even if you forget, you should be able to figure out, and the easier way to remember, the way we used to remember was, suppose there is a huge pipe and a hollow pipe, smaller pipes, inside that even smaller pipes, inside that even smaller pipes. So, it is kind of a barrel. Those of you who open a gun or something, you must have seen the barrels. Okay, it's like a barrel. Within a barrel there is another barrel, inside another barrel likewise. Or those of you have seen the Russian dolls, you know, those wooden stuff. So, inside one doll there is another doll, you open that doll, there is another doll. It's something like that. It's a very unique structure to look at, and especially when you look at in tissue culture dishes how these myotubes are forming myofibrils, it's a joy to see them. Okay. Now, coming back to the self assembly of these two things, where in the previous class we ended that they form something like this, a sarcomere like structure out here, which is called a sarcomere unit. Now, today we will talk about even further self assembly of these myosins and these actins. Okay. Essentially, if we talk about the structure of actin and myosin filaments, so that is what we are going to start today. Structure of actin and myosin filaments. Okay. So, talking about the myosin filaments, let us start with the myosin filaments. Myosin filaments are composed of numerous myosin molecules, okay, that are bundled together. So, there are multiple such molecules and they are bundled together like this, okay, like this, along with a globular protein head protruding out. So, that globular protein head is here. So, out here, this head is like this, which yesterday I just left it like this. So, the structure is more or less like this. So, if you look at this structure eventually forms something like this. This is that globular head. And so, the structure what I drew yesterday was something like this. Essentially, it has like this. I did not purposefully do that, it will be confusing. So, now I am just putting it together, and these are nothing but filamentous proteins. Okay. There are thick and thin segments which makes this structure, and each individual myosin molecule is made up of 2 heavy meromyosin chain. So, what you have 2 heavy, this is called meromyosin, just a terminology, meromyosin chain plus 4 light meromyosin chain. So, sum total there are 6 chains, meromyosin chain. The 2 heavy chain wound around each other to form the 2 heads at one end. So, there are actually there are 2 heads which are formed here like this, the head looks like this, and they are formed by these 2 meromyosin. So, this head, now I add one more information, that was the reason I did not tell you yesterday. So, this head is actually something like this. Okay. It is kind of a fork, okay, as if there is a flower with 2 petals. If you have to remember, it is something like this. Okay. This is how it looks like, whereas the 2 heavy chains wound together to form the 2 heads one end and a tail at the other. So, this is where the tail is, where they are attached. Fine. Each myosin head also contains 2 of the 4 light chains that make up the part of a cross bridge. So, these are split up into 2. Okay. So, what essentially these are doing? So, each myosin head, this is 2 and this is 2, each myosin head also contains 2 of the 4 light chains, which makes the part of the cross bridge. So, I will come later what is the cross bridge, what we are talking about the cross bridge. So, cross bridge essentially is, let me go back here, this zone, the zone what you see here, this is termed as the cross bridge. Okay. This is the zone which is called a cross bridge, and the movement here is like this, either sarcomere will shorten. So, the option is that sarcomere length, either it will become shortened or it will remain in a normal relaxed position, or it will remain in an extremely relaxed position. So, if you follow this drawing, so when it gets shortened there is maximum amount of interaction, but then these red lines move here when it is getting shortened, right, because you are shortening it, though there will be more cross bridge area, accept it. Whereas, if they go further away, then this red line will shift further outward. So, automatically the number of cross bridges, if the interaction is something like this. So, okay. So, if they are close into each other, this is where there is a shortening, there is a maximum amount of interaction among each other, and if they relax like this, there will be a minimum amount of interaction and there will be a zone of optimal interaction, right. So, not they are very close like this, which is shortened version, very shorter, or they are very relaxed, like kind of chilled out from on each other, or something like this. So, there are 3 possibilities. They may have a kind of, say for example, I say, let us give it a number. If I say there are 50 connections between these 2 pumps like this, okay, there are 50 something like this, connections like this. Okay. So, now or let us put it like this, there are 5 connections like this. Now, if we move away, there may be only 2 connections left, or if we are somewhere between, there will be 3 connections. So, 3 is somewhere in between 5 and 2, right. So, something like that. So, there is a change in length of the sarcomere they come too close, maximum interaction between the cross bridges, and cross bridge is essentially is this area out here. So, now let us correct this drawing little bit further, improvise this drawing like this. So, these heads are forked kind of thing. Okay. So, number of cross bridges decides the interaction between the two. And now, if you look at the actin filament, now coming back. So, we talked about the myosin filament, details of the myosin filament. And if you look at the actin filament is even much more interesting. The actin filament contains 3 proteins. Okay, let us put another color to this. Okay, once again. So, we talked about the myosin. Let me talk about the actin filaments. They form, actin contains 3 protein. One is actin itself. Then you have tropomyosin and troponin. Tropomyosin and troponin. Okay. It is made up of 2 helical strands that are loosely connected, made up of 2 helical strands which are loosely connected okay. 2 helical strands which are loosely connected. One strand composed of actin filament. On the other hand, the other strand consists of the tropomyosin. So, second strand is tropomyosin molecule. The tropomyosin strand covers the actin. So, the actin filament, if you look at it, actin has multiple sites. Okay. Actin, if you look at the actin filament, so this is something like this. Okay. So, you have actin, tropomyosin, then you have something called active sites on it. Let us put a different color out here. Active site, and then you have calcium binding site, which is, this is something new I am introducing now, called calcium binding site. So, I think at this stage you people need to know only 4 things. So, it has a actin filament, let us put a different color to this, which is in green, the actin filament, and then you have a tropomyosin filament, which is in blue, the tropomyosin, this one. Okay. And then you have an active site which is present out here. An active site is the site where it is interacting with the myosin filament. Okay. And then you have a calcium binding site, right. So, tropomyosin strand covers the active site under a normal condition. So, active site has certain binding pockets, okay. Those are covered by the tropomyosin molecule. It is something like it is like this. Okay. So, tropomyosin hides the active site. Okay. Now, a troponin molecule is attached to the tropomyosin molecule, and troponin is made up of, now there is another molecule which is present there, which I have not mentioned yet, which is the troponin. So, this particular molecule is attached to the tropomyosin. Okay. So, let us put another color to this, let us pick up this or maybe this color, okay. Something like this. Okay. So, there is a troponin molecule. So, the troponin molecule attached to the tropomyosin molecule, this troponin, and it has 3 binding sites. It has troponin A, which has affinity for actin, it binds to the actin. Then you have troponin C, which affinity for calcium ion, this binds to the calcium ion, and troponin T, which binds to the tropomyosin. Okay. So, now tropomyosin adheres to the troponin molecule. So, now again let us re-summarize it. Actin filament made up of actin 1, tropomyosin 2, and another molecule called troponin, which is the third one. Now, troponin interacts with the tropomyosin. Troponin has a binding site with actin, which is called A site, troponin A. Then troponin has a affinity for calcium ions, which is called troponin C, and troponin has a binding with tropomyosin, which is called T. Fine. And you have an active site out here, which is hidden by the tropomyosin molecule. Okay. At rest, if you see, there are no chemical reaction between actin and myosin filament, and the molecules are stretched without much resistance. But in the presence of calcium ions, so I showed you a calcium site, right? But now, in the suppose in this matrix, now let's get. Now, if you are introducing calcium, we will talk later from where calcium is coming. This is another very fundamental problem, we will come to that. Now, when you are introducing calcium ions, so say for example, this milieu is now filled with calcium. Okay. Under that situation, what will happen? Actin and myosin bind strongly to one another. It is believed that binding of calcium ion to troponin physically moves the troponin tropomyosin complex, so that the active site on the actin filaments are exposed. Okay, what does that mean? Let me put it together. Okay, one second. Okay. So, say for example, at rest, let us break down, talked about at rest, no chemical interaction between actin and myosin filament. All right. So, the muscle can be stretched without any resistance. So, muscle can be stretched. So, we are making statements now. Okay. S1, statement 1 can be stretched without WO, without resistance. Statement 1. Now, we are moving on to statement 2. Statement 2 is in the presence of calcium ion. Okay. In the presence of calcium ions. Okay. Now, calcium is introduced into the system. The actin and myosin bind strongly to one another. Actin and myosin binding happens. We will come later about all these bindings and the generation of the power stroke and all that stuff. Okay. Actin and myosin strongly bind to one another, and it is believed that the binding of calcium ion to troponin. So, coming back here, so the binding of calcium ion, I told you that troponin has a binding site for calcium. So, as soon as the calcium binds to the troponin, it exposes this active site. Okay. It exposed this active site, and by exposing the active site, just try to understand it first of all. Okay. So, this is the myosin head. all right. So, if this is the myosin and this is the myosin head which is present there, okay, something like this. On top of that you have the actin filaments. So, my right hand, this hand with this kind of, this is the this is the actin, and here you have the myosin head underneath. This is, so this is the myosin head, follow me, okay. This is the actin, fair enough. Now, under normal condition there is no interaction, so they are not touching each other, and I told you on this actin you have tropomyosin filaments, troponin filament, and an active site which is hidden by the tropomyosin, and you have this troponin molecule which can bind to calcium. Now, this is the normal interaction. At this stage you can stretch the muscle as much as you can because there is nothing to hold on to this, fair enough. Now, as soon as calcium comes into play, suppose it is raining, imagine the rains are the calcium. As soon as it rains, what will happen? The troponin will bind to the calcium and will remove or expose the active site on this actin molecule, and as soon as that active site is exposed, the myosin head binds to that exposed site. Now see, now this is the stage, stage 1, stage 2, like this is a bind, and after binding there is a reaction which takes place whereby there is an energy release which leads to the bending of this head like this, and during that head it moves this actin filament like this, it something like this. Does it make sense? Let me draw it for you because this is a fundamental concept you have to understand. So, you have this is your actin filament without any interaction, and this is your myosin head. There is no interaction, right. Now, stage 2, when there is an calcium ion coming. As soon as the calcium ion comes, on these actin filament there are sites which are supposed to bind to this is actin, okay, which are supposed to bind to the myosin filament. So, now this myosin head, this is myosin, binds to this active site. So, this is the zone of active site. If you remember in this picture here I told you the cross bridge. So, this is that zone where the cross bridge I am talking about. So, here the cross bridge, the binding takes place. So, some form of, let us put another color here. So, there is some form of binding happens here, and during this phase this head makes a movement like this. The head, look at my hand. So, head moves like this, myosin head moves like this. Once the myosin head moves like this, so what it does, if this is the actin on top of my hand, when it does so, it moves the actin along with it because it is attached. Now, imagine that there is a glue here. I have some glue, some sticky glue, and it just gets attached here. What it will do, if I pull my this hand, along with it, it is going to pull this one. So, what will happen is that while I am pulling this one, pulling this myosin filament, because its head is moving like this, its head is moving like this, it will pull the actin filament along with it, once again actin filament along with it, and this is a energy intensive process. This needs energy, okay, and this process is called the term is power stroke, and this is that first step of force generation. But then when I introduce force generation, I have left with you with lot of confusions from where this molecule came, right, from where calcium came. I have not told you from where calcium is coming, but this leads to something called a sliding. of actin and myosin on top of each other, and this is what I explained or what I started in the last class called sliding filament theory of muscle. What we will do from here, it is just the beginning of understanding these forces when they have a direct link to the nervous system. There will be a spillover now. Next week, from this power stroke, we will figure out the force generation curve of the muscle. Though I was supposed to finish in this week, but do not worry. Let us understand the basic concept once and for all. Once you understand this, I know rest of your life you will never have any confusion. You will build up the story. Okay. So next week what we will do, we will talk about how this individual power stroke. Now think of it for a minute while I drew this. So at individual level there are power strokes which are getting generated. Think of it, how beautiful it is. So there is a huge power stroke happening here, and that gets translated into here. There is a huge amount of force here, and eventually this whole thing moves with a force, and that's all start with molecular machines. And these are the real molecular machines, but there is a small, some nanonewton something force is generated there, but that eventually leads to what you see today, exercise, everything, what all we do, but it starts at the atomic resolution level. So I'll close in here today, and next week we will again take up from where we ended here, the power stroke. From power stroke we'll resume the story of muscle, and which will take us to the domain of neuroscience, which will help us to explain from where these calcium ions are coming. Okay. So apart from it, there is another very interesting thing what we'll be dealing with, which I haven't dealt, which will start in the next class, about excitation, contraction, coupling apparatus. Okay. Thank you.